Hi, everyone. My name is Naomi Sanchez, and welcome back to another episode of my podcast, Plantastic, a podcast where we discuss all things plant biology. On today's episode, we will be discussing stem development, going over plant anatomy, the importance of stems, and the way they get developed on a mechanistic level. Joining me today are two lovely plant biology experts who will now introduce themselves.
Hi guys, my name is Emalina Stepanian.
And I'm Kevin Deras-Guerra.
And now we can get into the content of the episode. So first thing I wanted to ask is to Emalina, and that was just to give a background on STEM development overall.
Perfect. So basically, stems play a major role in the lives of plants and animals. They serve as food, fuel, shelter, etc., and they're apparent in our everyday to day lives. And especially within our diets, a fun fact actually is that a major source of carbohydrates, which is potatoes, are a form of modified stems.
So what
Um,
exactly is a stem?
Stems are vertical structures that support the plant mechanically, conduct water and nutrients, and store food, and they come in a variety of lengths and shapes, and they can look woody, herbaceous, tall, short, thick, or thin. So anatomically, stems have two important structures. They have the nodes and the internodes, and nodes are where the leaves can attach to the stem, and internodes are the spaces between the nodes. And when we think of stems, there's basically two monocots and you have eudicots. And both of them have vascular bundles that consist of a xylem and phloem surrounded by ground tissue. In monocots, the vascular bundles are scattered throughout the stem tissue, and in eudicots, the vascular bundles are arranged in a ring. And the type of growth that stems have include intermediate growth, which basically just means that the cells will continue to grow throughout the plant's life cycle, and they don't really have like a halting period that they stop. Where it all begins is that the first rudiment of the young stem or shoot of an embryonic plant appears from the seed after the root was first protruded. The growing portion of the apex of the shoot is the terminal bud of the plant, and by the continued development of this bud and its adjacent tissues, the stem increases in height. As the stem continues to grow, lateral buds are produced that develop into lateral shoots, more or less resembling the parent stem, and these ultimately determine the branching of the plant. In trees, the lateral shoots develop into branches from which other lateral shoots called branchlets or twigs arise. The point at which a leaf diverges in axis from a stem is called the axil. A bud formed in the axil of a previously formed leaf is called an axillary bud, and it, like the leaves, is produced from the tissues of the stem. So the process of stem development in plants is broken up into two main groups. You have primary growth and you have secondary growth. The primary growth is where we're dealing with the apices or tips of the stem, and this happens when we're dealing with rapidly dividing cells in the apical meristems or in the shoot tip. And the apical bud has an influence on the overall growth of the stem, as well as a thing called apical dominance, which basically is diminished by the growth of the axillary buds that form along the sides of the stem. Secondary growth has to do with the growth of stem thickness due to the activity of the lateral meristems. This includes the vascular cambium, and in woody plants, the cork cambium. So the vascular cambium is located just outside the primary xylem and to the interior of the primary phloem. The cells of the vascular cambium divide and form secondary xylem to the inside and secondary phloem to the outside. The thickening of the stem that occurs in the secondary growth is due to the formation of secondary phloem and secondary xylem by the vascular cambium. So the cells of the secondary xylem contain lignin, which provides hardiness and strength.
Okay, so what about woody plants? How does that work?
So in woody plants, we actually have this thing called the core cambium, which is the outermost layer of the lateral meristem, and it produces bark, which contains a waxy substance known as the suberin that can repel water, and this bark will protect the plant against physical damage, and it helps reduce the water loss as well. And just to give a little overview, the hormones that are involved are gibberellins within stem elongation, and this is regulated by auxin. Okay, now I will be handing it over to my host, Naomi, who will be explaining the biological significance of stem development.
So stems are obviously central to any plant, and they play a major role. They serve as food, fuel, and shelter, and the way they get developed is important because it provides mechanical support. It aids with connecting all of the plant's organs. It provides transport for nutrients and water throughout the entire plant. And it's important for long distance cell to cell communication. And so when we're looking at a stem, we have the leaves and roots are obviously connected to it. And this is important because that's where all the water and nutrients are going up from the roots. And so we also see that sugars are leaving from the leaves to the rest of the plant, and stems are also important for supporting flowers and fruit in different plants. Stems are also a source of support for plants. So there are many plants that have their stems grow to specific lengths that allow them to ensure that they don't break due to wind or heavy rain conditions. Stem development is very important when it comes to photosynthesis because having evolved photosynthetic organs above ground are key to plants' success and thriving for as long as they have. And the taller a plant is able to grow, the closer it is to direct sunlight, which obviously aids in photosynthesis.
Great. And if we're thinking about it at the mechanistic level, Kevin, can you explain and talk a little bit about how hormones play a role in stem development?
Yeah, sure thing. So growth is rooted in the primary meristem. You may have heard of it as the shoot apical meristem, and that's affected by hormones that we've mentioned, such as cytokinins, auxins, and gibberellins. So primarily phytochrome stimulates growth toward red light, and that's going to help the plant know which direction to grow. This is called phototropism. It pushes the stem toward the direction of any sunlight. Cytokinin is going to promote cell division, and then gibberellins are going to promote elongation. And basically what that looks like is that the cell is going to expand in space between its cell wall. This is going to allow it to grow in length, and then cytokinin is going to encourage the cell to then cleave, and then it's going to form two daughter cells. Cytokinin can only divide cells in the presence of auxin, and that's particularly interesting because auxin is an inhibitor of gibberellin. So you have this cool system where auxins are inhibiting gibberellins, which would be promoting elongation, and then cytokinin can only split those cells after elongation in the presence of auxin. So auxin is kind of that balancing mediator that's allowing cytokinin and gibberellin to understand when it should elongate and when it should cut. And this comes into an interesting phenomenon that we mentioned, where you observe gravitotropism, which is what it sounds like, essentially the involvement of gravity in the development of stems. Because gravity is always acting on these plants, you'll have auxin, which pulls toward the bottom of the plant or wherever gravity is pulling in the direction of that plant. And since we know that auxin is an inhibitor of gibberellins, it's not going to allow for the stem to elongate in that direction. And what you're going to see is a bend toward the opposite end of gravity due to that pooling of auxins. Um, when you look at stem development in particular outside of the shoot apical meristem, you'll see these organ boundary genes, which are important control points in early stem growth. Stem elongation is what drives the bending and the general lengthening of the organ boundary genes. But then you have these proteins which are produced in the genome of those cells in the more central sections of the stem called DELA and BLH, which are involved in coordinating the patterning and the orientation of the growth.
Okay, that was a great explanation. I was also wondering, Kevin, if you could explain a little bit about how secondary meristems come into play for the development of stems.
Yeah, so as you mentioned before, girth is an important part of the stem development. It provides a lot of structural support, and so that's going to be maintained through secondary meristems. There are two types of secondary meristems. There's the vascular cambium and the cork cambium. As Emalina had mentioned earlier, the cork cambium is primarily involved in woody species of plants, such as trees. And so focusing particularly on the vascular cambium, we're looking at a system that inlays xylem and phloem into the stem, and those are going to be our main transporting systems for getting nutrients up and down the length of the plant. As the xylem and phloem are inlaid, the diameter of the stem increases, and this entire system is going to be mediated by auxin. In the cork cambium, you're going to find the development of bark, which is a rigid outer layer where the epidermis is replaced by cork. This is generally laid between the epidermis and the phloem, and you'll observe this development of annual growth rings where the vascular cambium gives rise to the growth rings. It lays xylem and phloem in the spring growing season. The xylem have a large internal diameter. Their primary cell walls are not exclusively thickened, and this is known as early wood or spring wood. During the fall, you'll see a secondary xylem system that develops in thickened cell walls, and this forms late wood or autumn wood, which is denser than early wood. After this, you're going to see the development of the cork, which is done by the cork cambium in those woody species. But outside of those woody species, you will generally see this continued development of the xylem and phloem in the different sections of the seasons and using that alternating system of primary and secondary xylem.
Okay, that's about all we're going to cover on today's episode. Thank you, Emalina and Kevin, so much for joining me, and thank you to all the listeners. I will see you all next week where we're going to be discussing circadian rhythms in plants. Thank you so much.
Thank you.